Hello everyone, welcome back to our channel. I'm Dr. Ashmita. In this video, we'll continue with the series of G-protein coupled receptors. In our previous video, we have discussed about the mechanism of GPCR signaling, that is, how upon this ligand binding, the receptor gets activated and it leads to the activation of a G-alpha subunit. And this active G-alpha subunit, it will dissociate from its beta-gamma dimer, and the active G-alpha subunit will choose pathways like stimulatory, inhibitory, Q or 12-13. In the previous video, we have emphasized much on this G stimulatory pathway, G-alpha stimulatory pathway. Now, in this video, we'll study about this inhibitory pathway. So, before understanding the inhibitory pathway, let's again revise the G-alpha stimulatory pathway, because to have understanding of inhibitory, we need to know the stimulatory pathway clearly, because inhibitory is the exact opposite of the stimulatory pathway. So let's revise. This is the GPCR. Upon this ligand binding, the receptor will convey this signal, extracellular signal, to the G-alpha subunit of the G protein complex. The alpha subunit will dissociate from the beta-gamma dimer, and this active G-alpha subunit, now it will stimulate this, it will take on this journey of G-alpha stimulatory pathway depending on the ligand and the receptor. Whenever the stimulatory pathway gets activated, it will lead to activation of effector called adenylyl cyclase. So this is the effector, adenylyl cyclase. It will ultimately lead to generation of secondary messengers like cyclic AMP. Cyclic AMP is a secondary messenger which act on a protein kinase A. It will activate protein kinase A. The protein kinase A will ultimately act on the target proteins. So, from the active G-alpha subunit, the stimulatory pathway got activated. So, G-alpha S will activate effector. Number one step, effector. From the effector, cyclic AMP, secondary messengers. From the secondary messengers is the protein kinase A. Protein kinase A will finally act on target proteins to produce this ultimate final cellular response. This is the stimulatory pathway. Now coming to the inhibitory pathway, which is the exact opposite of the stimulatory pathway, and as I have said before, the pathway, the G-alpha stimulatory or inhibitory, the pathway is chosen depending on the ligand and the receptor. So the ligands, the GP coupled receptors linked to this inhibitory pathway are the alpha-2 adrenergic receptors, M2 muscarinic receptors, D2 dopamine receptors, mu opioid receptors, and the somatostatin receptors. These 1, 2, 3, 4, and 5. These are the few examples of receptors that, upon getting activated, will lead to the G inhibitory pathway. Let's study the inhibitory pathway. So, this G alpha inhibitory pathway, inhibitory subunit, it got activated. It will inhibit the effector. The number one step of the G alpha S is this adenylyl cyclase effector. This effector will be inhibited. It has a negative impact. Because there is no effector, there will be no generation of the secondary messengers, that is the cyclic AMP will go down, because cyclic AMP is produced from ATP with the help of this enzyme adenylyl cyclase. Adenylyl cyclase will convert ATP to cyclic AMP. When there is no adenylyl cyclase, there is no cyclic AMP. When there is no cyclic AMP, there is no activation of protein kinase A. The protein kinase A activity goes down. So its effect on the target proteins is also gone down. It will also go down. Target proteins are not activated. So final cellular response is not produced. This final cellular response is not produced. So all the opposite actions of the G alpha stimulatory, all opposite to the G alpha stimulatory is equal to the G alpha inhibitory. So once again, let's revise. Starting from whenever this inhibitory got activated, the effector, the first step, effector got inactivated. It will inhibit it. Because of that, cyclic AMP, secondary messenger, is not produced. When no secondary messengers, protein kinase is not activated. So there's no like activation of target proteins, no activation of target proteins, no final result. This is about the inhibitory pathway. Say, for example, consider this alpha-2 adrenergic receptor. Upon activation, it will choose this inhibitory pathway. That means the neurotransmitter, the final output, norepinephrine, is the release is decreased. This is an example of this inhibitory pathway. And this signal, inhibitory signal, it also needs a termination. So termination is brought about by GTPase. GTPase, we have studied the mechanism here. The G alpha subunit has two domains, GTPase domain and the helical domain. Yes, the GTPase domain will cause hydrolysis. It will cause the hydrolysis of GTP to GDP. That means GTP means this active form of G alpha, GDP means it is inactive. So it will cause hydrolysis of GTP. That means it will convert the GTP back to GDP. That means it will convert the active form of G alpha to inactive. So here GTP is converted to GDP. GTP means it is active. It is being converted to inactive form. Inactive GDP means this subunit is inactive. There's no signaling happening. Signal terminated. Other is phosphodiesterases. They degrade cyclic AMP. Here at this step. At this step. Cyclic AMP is degraded by phosphodiesterases. Phosphodiesterase enzyme will degrade the cyclic AMP. No cyclic AMP, all the steps are gone. There's no signal occurring there. And we have said this alpha subunit, it will dissociate from the beta gamma dimer. So here this one at one side this alpha signaling is occurring, and on the other side the beta gamma signal, it will function as a single molecule, it will function as a single unit. So this beta gamma dimer also has its independent pathway, signaling pathway. So this signaling, it will act on channels. Channels like it will open potassium channels. Because of opening of potassium channels, it will cause hyperpolarization. Hyperpolarization means there's decreased excitability in the heart muscles. That means it is acting on the M2 muscarinic receptors and causing decreased heart rate. And it will close the voltage gated calcium channels. When the calcium channels are closed, the calcium influx gets reduced. That means this decreased neurotransmitter release in the neurons, it will lead to analgesia. These are the two functions of signaling pathway of the beta gamma dimer. It will result in decreased heart rate via M2 muscarinic receptors in the heart, and also it will lead to decreased calcium influx, means decreased neurotransmitter release. It will lead to analgesia. And thirdly, it will activate phospholipase C beta. The phospholipase C beta, this we are going to study in this G alpha Q pathway. In the G alpha Q pathway, this we are going to study about this phospholipase C beta, which is present just under the plasma membrane. So we'll discuss it under the G alpha Q pathway, this step. So this is about the G inhibitory step, the G inhibitory pathway of the GPCR signaling. In our next video, we'll continue with the G alpha Q. So if you find this video useful, please like, share and subscribe to our channel. Thank you.